All right. So 1 second. Yeah. So yes, so from today onwards, we will discuss about flow cytometer. Okay. So what is this flow cytometer that we will see? So flow cytometer. So we will discuss in detail about flow cytometer. So let me take one white screen. So let me tell you some significance of a flow cytometer. So here, I hope you can see my whiteboard. So let me tell you about the cells. Okay. So in a cell, so we have the cell. Okay. So far in the first we have seen the cell has many antigens, right? So cell has many antigen receptors. So cell has many receptor like this. Okay. So I'm showing receptors in various types. Okay. So these are various types of cellular proteins. Now the cellular proteins can be detected by two means. One is by using antibodies. So with the help of antibodies, we can target the proteins. That is what we have seen in immunohistochemistry. So in the immunohistochemistry, we are targeting the proteins with the help of antibody, and this antibody is conjugated with some enzyme. Okay, it is conjugated with some enzyme. Now this enzyme will give some color. It will react with a substrate and it will give color. So this is how immunohistochemistry has how working. Okay, in immunohistochemistry, we are using antibodies to target the specific antigen on the surface of cell with the help of conjugated enzymes and their substrates. So the substrates will give color, and that is how we are visualizing the cells. So this is the how immunohistochemistry is working. So I'm writing IHC here. So this is the working principle of IHC. So this is how we can able to visualize the cells. Okay, that specific protein can be visualized in immunohistochemistry. Then in the second approach, now we have the cell and cell has enzymes. Okay, so in the second thing, now we need to detect these enzymes, right? So cell has enzymes, and detection of enzymes also has crucial clinical importance, right? So because many cells has metabolism, and metabolism is extensively mediated by enzymes. So enzymes detection is also important, and these enzymes can be detected in enzyme histochemistry. So we have many methods, like a directly substrate can react with the enzyme and it will develop color, or it will be secondarily, it will be reacted with some salts, diazosodium salt. Okay, so this is a salt. So with the help of salt precipitation, we can able to visualize the enzymes within the tissues. So this is about enzyme histochemistry. So this is enzyme histochemistry. Now, both techniques are fine. So in case of immunohistochemistry, we are detecting cellular proteins nicely. And in case of enzyme histochemistry, we are detecting enzymes within the tissue. And in your syllabus, we have third technique. The third technique is detection of DNA and RNA within the cell. Okay, that is called in situ hybridization technology. So in situ hybridization technology, what happens is we can able to detect DNA and RNA. Okay, the cellular genome. So this is genome. Okay, so this is a DNA. Now this DNA can be detected in fish. Okay, fish stands for fluorescent in situ hybridization. So in situ hybridization technology, IES. Okay, in situ hybridization technology. So in situ hybridization technology, the antibodies will be will be targeted to the genome of the cell. Keep in mind. Okay, antibodies are targeted to the genome of the cell. So now these antibodies either they will bind to the DNA or RNA of the cell. Okay, so by this fish technology or fluorescent in situ technology, we can able to detect the DNA or RNA, the genome of the cell, right? So these are the three major fields of advanced histochemistry, and this is the three important units you have in your syllabus. Okay, you have immunohistochemistry, detection of cellular proteins or antigens with the help of antibodies, and second one is enzyme histochemistry. Here we are detecting cellular enzymes by using some substrates. Then we also have a specialized advanced technique called fish. So in fish in situ hybridization, here we are detecting the DNA or RNA with the help of some molecular probes. So here we rather than saying antibodies, we will say probes. Okay, we will have specific probes. That probes will bind to either DNA or RNA, and these probes are conjugated with some enzymes or with some radioisotopes or fluorescing components, and those fluorescing components will give us color. Okay, so these are the three major branches of advanced histochemistry. So this is it. Now, we have there are unlimited practical applications of these three branches actually, because let me tell you one thing, like malignancies. So if we take cancers, we will get blood cancers. Let us focus on blood cancer. So in blood cells, we have white blood cells, right? In white blood cells, we have granulocytes and agranulocytes. So if you go to agranulocytes, we have monocytes and lymphocytes, right? Even in lymphocytes, we have two types of cells. We have T cells and B cells. So there are some cancers that will cause due to T cells abnormal growth. Okay, that is called T cell mediated cancers, and we have B cell mediated cancers. So myeloid cancers and lymphoid cancers. So if it is a lymphocyte cancer, it is called lymphoid cancers. If it is monocyte cancers, they are called myeloid cancers. Even here we have acute and chronic conditions, acute and lymphoid cancers, chronic lymphoid leukemia. So we have multiples of cancers. These cancers are because of either T cells or B cells. So the T cell and B cell cellular proteins, CD4 and CD8, CD7, CD6, CD8. So all these cellular proteins can be detected in immunohistochemistry. So all types of cancers can be detected by immunohistochemistry, whereas enzyme histochemistry or enzyme histochemistry can able to detect enzymal dysfunctions, and even in FISH, in fluorescent in situ hybridization, we can able to detect any cancer genes within the cells. So there are broader applications of these three fields in histochemistry, right? Now, the problem with these three techniques is they are really time-consuming procedures. It will take almost 2 to 3 days to get report. And also all these three tests are very costly. And moreover, it is all about our time-consuming. It will take at least 3 days to get reports from these three techniques. Because in all these three techniques, we need to subject the tissue to tissue processing techniques. So tissue processing, we already know that it will take so much time to process a tissue. So because of this tissue processing problem, it will take more time to get the reports out of these three techniques. Okay. Now, is there any advanced technique that we can able to adopt to fasten this process? Is there any shortcut approach to detect all these three things within a fraction of seconds? Is there any such fast advanced technique? Okay, so if we have such technique, it will be a breakthrough. It will be a huge advantage in the field of histochemistry. We can able to detect anything in the matter of seconds from the patient's data. Okay, rather than waiting for 3 days, we can able to generate report within 3 minutes. Is there any such technique to get to diagnose these problems or these disorders within 3 minutes? Okay, so that is how we got a new technique. Okay, so now I will discuss this new technique. This new technique is called flow cytometer. Okay, this technique is called flow cytometry. So I'm writing here flow cytometry. Flow cytometry. So it is a cell counting procedure. So flow cytometry. Okay, so this is called flow cytometry. So the idea in flow cytometry is, so I will tell you a few interesting things, then we can see every other things. So in flow cytometry, I can detect cells in the motion. Okay, so cells will be passed. I will have one laser like this, one tube. So from this tube, I narrow this tube so much small hole that only single cell will pass through this tube. Okay, only single cells will pass through this tube. Okay, and the real interesting thing is now the cells flowing at a rate of 20,000 cells per second. Okay, sorry, 2000 cells per second. So they are flowing at a speed of 2000 cells per second. Okay, now is it possible to count 2000 cells per second? Is it possible or not? So it is really challenging, but our flow cytometry can able to detect 2000 cells per second. That is the major advantage of our flow cytometry. It can able to precisely count the cells very fastly. Now I want to tell few more parameters of this flow cytometry. So this is the meaning of flow cytometer. Okay, flow means the cells are in constant motion. So cyto, cells, metry, to record. It is the recording of cells that are in motion is called as flow cytometry. Okay. Now, what are the capabilities of flow cytometry? What parameters does our flow cytometer can detect? So first one will be counting. So cell count we will get. So count of cells we will get automatically. So the number of cells passing through per second will be count. Okay. So we got the data of counting of cells. Now we can also able to detect the size of the cell. Out of these 2000 cells, how we can able to detect the sizes of individual cells. Okay, so we can able to detect the size of the cells. Okay, so I'm writing size. So the cell size data we will get. And now the cellular complexity can be measured. Cellular complexity means whether they are granulocytes or not, whether they are agranulocytes. If they are granulocytes, are they monocytes, are they lymphocytes, are they neutrophils, that we can detect. If they are agranulocytes, are they monocytes or lymphocytes. So we can able to even check the complexity of cells. Okay, so complexity, I'm writing complexity of cells. So we will get the data of complexity of cells. Okay, so these three looking very simple. So we can able to count the cells, we can able to identify the size of the cells, we can able to identify the complexity of the cells. Now we can even able to detect cellular proteins as we are checking previously here. So I said that here we can able to detect the cellular antigens. So even cellular antigens can be detected. So cellular antigens can be detected with the flow cytometer. Now, how we will detect this antigens means we will label the cells with various types of fluorescent labeled antibodies. Okay. Now, in a single cell, almost 20 antigens can be detected. That means I can label a single cell with 20 antibodies. Now I will get 20 antigen data from a single cell, whether the cell has CD4 receptor or not, whether the cell has HLA receptor or not, whether the cell has other 20 proteins I can detect in the same time. So within 1 second, I will get, I can able to count 2000 cells with their size, their complexity, their my specific antigens. So I can even detect specific antigens in flow cytometer. Now, not only this, we have even more applications of flow cytometer. The other application is I can able to sort the cells, sorting of cells. You know what is this sorting means? Sorting means, see, out of this 20,000, I want to detect one particular CD. My antigen of choice is CD24. Let me say CD24. Okay. So now, if any CD24, so here I got CD24 cell. So my computer detected CD24 cell. So when it is in this motion, when it is detecting by this flow cytometer, if it is detected CD24 cell, this CD24 cell will filtered. It will be filtered into a basket. So now I can get only the specific CD24 cell into my basket. So I can able to even sort the cells. Sorting of cells is possible by this flow cytometer. So this has a very important application in the field of this fertility centers. Okay, now because I want to take the best sperm out of my donor. Okay, the sperm that has the high chance of getting high chance of getting pregnancy. Okay, so now out of this 120 million sperms in 1 ml of semen, I want only that one particular sperm which has the capability. So that can be identified, that can be filtered, that can be sorted, and that can be utilized for IVF, in vitro fertilization. So you just imagine the potential applications of our flow cytometry. So it can able to sort the cells. Okay. And also there is some software gimmicks can be done with this flow cytometer, that is called gating. Okay, that concept is called gating of cells. What is this gating means? See, just by running flow cytometer for 1 minute, okay, just by running flow cytometer for 1 minute, how much data we can able to generate? You know, we will get a data of how many 2000 cells. Let me do that calculation. So 2000 cells into 60 seconds. Absolutely, we will get 1,20,000 cells data in a minute. So we will get 1,20,000 cell data. You just imagine how potential it is. So by running flow cytometer for 1 minute, I will end up with getting data equivalent of 1 lakh cells. So 1 lakh cells data I will get it. In this 1 lakh cells, I can able to know how their count, their size, their complexity, my desired antigens, and I can even able to remove the specific cell that I want from flow cytometer. So illustrating, visualizing all these cells and their parameters is called as gating. So gating is just like a modified PowerPoint presentation. The statistical representation of the cells, the cellular data statistical representation can be given by this gating approach. So these are the potential properties of our flow cytometer. Flow cytometer can able to detect cells in motion at a speed of 2000 cells per second. It can able to count the cells. It can able to give the data of the size of the cell, the complexity of the cell, the presence or absence of our desired antigens or surface protein receptors on the cells. It can able to remove, it can able to capture, filter, or sort the desired cells. It can even able to give us the visual representation of the all cellular subsets data in a computer. So all these are possible by our flow cytometer. So how does this flow cytometer able to count the cell? How flow cytometer able to measure the size, complexity, antigens? All these things we will discuss in the coming weeks. Okay. So how is it possible? That I will discuss now. Okay. So I'm going to the next slide. Another white slide. By the way, is everyone listening to me, students? Are you following my lecture? Yes, Damani. Yes, thank you. So I will go to next slide. Now I will show you the basic principle of this flow cytometer. So flow cytometer has the following components. Okay, it has fluids. So I'm writing components of flow cytometer. So it has a fluids, FLUIDS. It has a fluids component. It has a lasers. It has lasers. Okay, so these lasers are very important. It has lasers. Then it has optics, some optical things. It has optics. Okay, it has optics. Then of course it has electronics. Of course it has electronic components. Include computer, everything. Okay, electronics. Electronics. Then apart from it, we need sample labeling techniques. Sample labels also required. So sample labels are also required here. So now let me tell you how this flow cytometer works. So we got a laminar flow of cells. This laminar flow of cells means cells which are going in single line, single profile of cells. Okay. So for this purpose, we will use a very, very narrow tube. Okay. This tube will have a apparatus, a small hole. That hole will be the it will be the size of the smallest cell. Okay, like thousand times smaller than a width of your head. That much small apparatus will be there. Okay, so we have a tube, and this tube is filled with some fluid. So this tube has filled with some fluid. So let me keep that fluid here. So this tube has some fluid. So I'm just drawing some fluid. Okay, assume this is a fluid. So it has a fluid. Fluid is very important in flow cytometer. Now In this fluid, the cells are suspended. So I'm drawing cells here. So cells are suspended in the fluid. Okay. Randomly cells are suspended. Now, these cells will start coming outside this narrow tube in a single cell. So only one cell can able to escape. So this is called laminar flow of cells. So laminar flow of cells started going outside. Then now, now I will focus a laser. So let me take one laser here. So this is a laser. So I took a laser gun. Okay. So this laser gun, what it will do? It will start giving out the laser. So now the laser light has been focused onto the cells. So for this focusing of laser light, we will use some optics. As I stated, we require some optics. So I used some optic, some mirror to focus the laser light onto the cell. Now, you just keep remember on the next, on the opposite side, I will keep a detector. So here I'm keeping a detector. Let me take detector in another color. So here I kept a detector. So let me draw a detector like this. So this is the detector. Okay. So I kept a detector. Now listen, students, very important. Now I know the intensity of light that I transmitted onto the cell. Say for example, this intensity is around 100 lumens. Let us assume 100 lumens is the intensity of the light when I focused onto the cell. Now, just opposite to the cell, I kept a detector. If any cell is there, what will happen? The cell will block the light. Okay, the cell will block the light. If after blocking, the amount of light that I received in the detector, so I'm just showing the leftover light. So the amount of light that I detected here is 90 lumens. Let me show you 90 lumens. So here I got only 90 lumens of light. Why the light has been decreased? Why the light intensity has been decreased? The reason is 10 lumens of light has been blocked by the cell. That's why I detected only 90 lumens of light. So this is called forward scatter. Okay, the light that here we have two scattering. So I'm writing here. I will write here. So we have two types of scattering. Light scattering, SCATTRING. So this light scattering are of two types. So I'm writing here. So one is forward scattering, FS. So I have forward scattering, okay, and other one is side scattering. So I will discuss side scattering later. First, let us see what is this forward scattering. So now the light has focused on cell and cell absorbed some light and some light has been scattered. This is called forward scattered light. So this is FS light. Forward scattered light is 90 lumens. So forward scattered light will give the size of the cell. Let us assume the cell is somewhat larger. So a very large cell passed via laser light. So if a very large cell passed, what happens? Then the forward scattered light will be decreased. So most of the light will be obscured by the cell. So the amount of light I will receive here will be only 60 lumens. So 60 lumens for large cell. So this is big cell. This is small cell. So just by measuring the forward scattered light, I'm getting the data of the size of the cell. So the cell size will can be measured with the help of forward scatter. So the size of the cell can be measured by the forward scattered light. By measuring the intensity of the forward scattered light, I can able to indirectly measure the size of the cell. You got my point, right? If the cell is very small, what happens? Let me show you another scenario. So cell is very small. If the cell is very small, very less light will be absorbed. So most of the light will be passed. Forward scattering will be more. So in that scenario, the forward scatter lumens will be around 98. So 98 lumens indicates that the cell is small. So this is the very small cell. I'm writing VS, very small cell. So by measuring the forward scatter light, I can able to see the size of the cell. So successfully now we understood how the size of the cell is counted in the flow cytometer. Now keep in mind, students, here I can able to count 2000 cells per second. I can able to count the size of 2000 cells every second. So you just imagine how complicated this flow cytometer is. It will cost around 1 crore to get this flow cytometer, but it has very potential applications. Okay, so this is all about the forward scatter. Now the second one is side scattered. So let me take two cells here, so you will get some idea. Let me take two cells. So I'm taking one cell here. Okay, so this is a basophil. I'm taking a basophil. So we know what is a basophil, right? Basophil has many basophilic granules. So let me take. So these are the granules in the basophil, and other one is I'm taking monocyte. So here I'm taking a monocyte. Monocyte don't have any granulocytes. So this is a granulocyte. So this is a granulocyte, and this is a granulocyte. Monocyte is a granulocyte. So I'm writing M here. So you will understand this is a monocyte, and this is a basophil. This is a granulocyte. This is a granulocyte. Now, you just imagine like this. Imagine oh, okay, you have two types of saris. Okay, woman has we have two women. Two women has two different types of saris. The first sari, she didn't have any. It's a plain sari. So plain sari won't have any glittering, that shiny effect. But the second sari has this mirrors. Small mirrors are there for the second sari. So like designer saris, fashion saris. Okay, so this designer saree has small mirror glitters, so they will be shiny. That means the second saree has more granulated. The second saree has many these small mirrors. Let us assume this mirrors like a granules. So the same idea. Okay, so let me tell you what happens here. If a granulocyte, is is moving through a flow cytometer, granulocyte will give more scattered light, side scattered light. So detection of more side scattered light indicates. So this is side scattered light. Side scattered light. Okay, SSC, SS actually, side scattered light. So if we are getting more side scatter light, that indicates it is a more complicated cell. Maybe it is a granulocyte. So only granulocyte will provide us the side scattered light. So detection of side scattered light will give the complexity of the cell. If a monocyte passing, it won't get any side scattering. So absence of side scattering indicates monocyte. Presence of side scattering indicates granulocyte. And the intensity of side scattering indicates various types of granulocyte. Say for example, neutrophils has 80 granules, basophil has 95 granules, eosinophil has around 85 granules. So depending upon by measuring the side scattered light, we can able to understand the complexity of the cell. So side scattered light will give the complexity of the cell. It will give the data of complexity. So whether it is a granulocyte or agranulocyte can be detected with the help of measuring the side scattered light. So simultaneously, a flow cytometer is measuring the count of the cell, the size of the cell by forward scattered light, and it is also simultaneously measuring the side scattered light to give us a clue of the complexity of the cell. You got it right? So these are the main components of our flow cytometer. It has lasers, it has fluids, it has detectors. Detectors are of two types: forward scattered light detector and side scattered light detector. Forward scattered light detector will detect the size of the cell, whereas the side scattered light detector will detect the complexity of the cell. Okay, so so this is one thing. Now I will show you a graph. Okay, so if this is called gating in flow cytometer. So in this graph, this bottom will be the forward scattered light. So this is a forward scattered light graph, and this side one is side scattered light. So here this is x-axis and y-axis. Okay, so 0, 1, 2, 3, 1, 2, 3. Okay, 1, 2, 3. Now let me check forward scatter. So if the cell is very small, we will get a very high light. So small cell will have here, the lighting will be here. We will get a graph here. And if it is a large cell, we will get here. So by measuring the forward scatter light, we will get the size of the cell. And if the cell is more complicated, then if it is a granular site, we will get around here. So here. So this is a small cell with more granulated. If we are getting around here, it indicates it is a large cell. Since it is here, it is a large cell and it is also complicated. So we are getting two data. So this will be a large cell with the granulated, means this will be eosinophil. This is a small cell and agranulated. It might be small lymphocyte. It might be small lymphocyte. And here, semi-granulated, less granulated but big cell. So less granulated big cell, it might be two types. It can be either monocyte or actually there is minor granulocytes. So it cannot be monocyte. It might be also a neutrophil. So we need further confirmation with other technique. Okay. So this is called gating of cells. So this is how we will get data in the graph. Okay. We got the forward scattered data and we got the side scattered data. So by plotting this graph, we can able to understand this one. So now I will show you the first slide. Okay, then you will get it what is flow cytometer. So in the first slide, I kept this one. See, so here we can clearly see a fluid system. So these fluids are releasing cells into a single profile. So cells are falling in a uniform. Okay, single cell is falling, and we have a laser. So this laser is giving two types of scattered light. This is forward scattered light, and this is side scattered light. So forward scattered light giving us the data of size of the cell, whereas the forward scatter light giving size of the cell, whereas side scatter light giving the complexity of the cell. So here we got the two data in the graph. So we got forward scatter data and side scatter data. So forward scattered, see, no, these cells don't have any granules. So that is why they're very small. They're at the bottom, but they're large. So large cell, no granules. Definitely there might be monocytes. So the bottom cells might be mostly monocytes. And here, these are granulocytes. They are large and they have granules. So granules and large cells might be granulocytes. So this is called gating in flow cytometer. Now, even within a granulocyte, we can further differentiate whether it's a neutrophil, basophil, eosinophil. Even in a neutrophil, we can further differentiate the type of antigen we want within the cell. So by labeling them with some fluorescein dyes, that I will discuss in tomorrow's lecture. Okay, so this is the brief summary of a flow cytometer. So flow cytometer is a device that will detect cells in motion at a speed of 2000 cells per second. It will give a simultaneous data of counting size of the cell, complexity of the cell, proteins or receptors present on the surface of the cell, and it can even able to gauge or filter the type of cell we want. Okay, that is called FACS, fluorescein activated cell sorting. That I will discuss in the next coming sessions. So this is briefly about flow cytometer, and let us see the applications of flow cytometer. So flow cytometer is very useful to detect various types of cancers, to detect minimal residual disease concentration in the body, to detect lymphocyte subsets in case of HIV, to detect the efficacy of cancer chemotherapy, to detect reticulocyte enumeration, cell functioning analysis, applications in organ transplantation. Actually, there are almost 200 applications for flow cytometer students. Almost 200 applications. You just understand the potential clinical significance in the diagnostic laboratory of flow cytometer. So flow cytometer is one of the